the stuff again. So just to remind you, these are the terms that we use, right? And these actually, for the lab, are the main ways that we refer to this. So don't always be a little concerned about saying viruses. We actually usually almost always mean VLPs. That's that basically physical view of the virus that is virion is what we're talking about, or something that's packaged like a virion, right? So it has a certain density and a gradient, it goes through certain filter sizes, etc. It's like a very classical thing. And that's what you count under the microscope, and we actually have no idea. Did anybody read the Stewart, from Greg Stewart's lab about the fact that we may be missing about half the viruses in the ocean? Did you guys read that paper? So it could be that we're not counting the small, especially the RNA ones, and we're not seeing them. And so they estimate that there may be as many of them as there are of the DNA ones, just so you know. So the diversity may be even a lot higher.
You can see them and you can't count them themselves.
What?
I mean, you can see them and you can't count those guys. Allison had them on a slide and she was like, what about all those little dudes? Like,
Yeah, it's not clear that we can count them.
No, I mean, you can't.
Yeah.
You can't on the algorithm, you just physically can't count them.
Yeah, even by eye. And Greg actually worked on that one when we were in Farouk's lab, because he did all those like initial studies, like showing where they came out in the pulse field gels and all that sort of crap. And so he really did actually look for them at the time. So this paper, you guys should, those of you that care about it, you should go look, because they did a pretty good job of extracting different nucleic acids and then quantifying how much they had. And they think that there's actually maybe half of what's out there we're missing right at the moment.
But don't our stains also only work on DNA?
What?
Don't our stains also only work on DNA, like the minor groove? So
Not
RNA.
all of them. Not all the stains. And we've tried, we being Maya and I, and probably Linda was involved, tried the RNA stains, the cyber stuff for RNA, and never saw much with those.
But technically, cyber labels both.
Only if it's got a secondary structure for RNA, right? I thought it had to have a groove for the cyber gold.
Or if it's double-stranded.
I think, don't quote me, but I thought that's one of the reasons in your paper that it works. We should look at that.
Yeah.
But cyber, I don't think cyber gold.
Cyber gold was just nucleic acid.
Well, we should look and see if it, so we haven't seen them,
But what
basically.
about with the TEM?
So the TM, it must be the shape and stuff is really hard to tell, because remember, most RNA viruses just look like schmutz. So if you don't culture them, they don't really look like much, right? I mean, they're basically just something that's wrapped up in some little protein coat. All right, so for a long time, we've been basically using microbes to being archaea and bacteria together, because usually we don't know what they are. And then, of course, remember the different life cycles. So you guys are all clear on this, right? I don't have to go through that. All right, and then as the background for this is basically just off a talk that I gave at in Portland. How do we know this?
Extrapolation.
From what?
Paragraphs.
From what? What's the literature?
See what?
No. What's it? Come on. You're
Oh,
a postdoc.
bacterial counts.
Bacterial counts. What's the reference? Guys, you're pissing me off. Right? I told you that you had to know that last time. Whitman. Right? Whitman did a paper called the Unseen Majority. Right? It is one of the most important papers in our field, so you better know it. Right? 1998. And from that, he calculates something like 10 to the 30th microbes out there, and we just say that there's about 10 times as many viruses, because that's roughly, approximately what we find out. Okay. This, how do we guesstimate that?
The fact stuff?
What?
The fact stuff?
Yeah, basically out of facts and kind of extrapolate. There's two ways. There's an argument in a cell preview paper that I make about, that's from 2003, where I calculated from facts and I calculated just from direct counts combined with average number of morphs. Okay, and you get about the same number. So there's something like that, and that's of course how you would define species. All right. Okay, so how do we know this? What are examples?
Kingston.
No, Thingstad wrote it.
Yeah, he
Thingstad
just
never
took it.
tested it.
Oh, you talking about how we tested it? Because there was the salton sea ponds and stuff like that.
Yeah,
With your
that might
strains
be the
underlined.
most complete stat
Mhm.
of that. But you can also see it all over the place in genomes too. Remember, you can see the places where they've, they changed the outer membranes and stuff like that. In fact,
Sludge.
yeah, exactly, the
Mhm.
sludge system, the cap system in particular. Linda actually is running into one, which is a very famous one from coli, which is the tone system.
What is it called?
TON, tone system, and that's T1 attacking coli, and what it does is it downregulates the ton. Anything that survives a T1 infection doesn't have it sticking up there. Ton and tol are those genes. Okay. All right. So we kind of see this, and so remember, this is basically the ecology, and this is basically the evolution. They only split them because they like to argue with each other. What do we call this in ecology?
Lockup Volterra.
Locke Volterra, for the most part, right? So this is Locke Volterra, and if you go and look at basically, Peter actually has a good way of explaining the rank abundance curves we see in natural communities using a Locke Volterra model that has an interaction term, and what that means is that things are probably happening on particles. Okay, and that might be important for your stuff eventually. We'll talk about gels in the ocean and stuff like that. Okay, all right. How do we know that we control the number of microbes?
Because they haven't overgrown everything.
What?
They haven't overgrown the whole world.
No. Come on, what's the experiment?
Filtered
No.
seawater on the corals and everything, right?
No, there's no coral involved here.
It's just seawater.
This is Wilcox,
Yeah.
Right,
it's the
okay.
seawater,
and he pulls it into
filtered,
different
unfiltered,
pieces,
with the
right?
DNIM and
Right, guys, you gotta memorize this shit, right? Because you have to know that. These are the experiments that tell us how we know this, all right? All right, how do we extrapolate this? That most of the genetic diversity on the planet is viral,
Unknowns.
right? Basically, that we know that we have more of them, and then we know that essentially it's all unknown, and it continues to remain unknown, right? Which is a little freaky. And I don't know exactly how to deal with that right at the moment, because our curve is still like this. Everybody else's curve is like this. They're not finding any more sequence space out there. Okay. All right, and then we know this. What's the experiment? There are basically two lines of evidence that are moving DNA around the world.
Find same genes everywhere.
Exactly, right? So that's our lab and Suttle's lab for the most part. Oh, and a little bit from the Pittsburgh group, Hatfull and Hendricks. Okay, what's the other one? The direct test of it.
they sequence the different oceans. Is that the same one?
Yeah, that's the same idea. No, exactly, Angley's stuff, right? It actually is this experiment, but done by Amiko, remember? And so you could take you could take the viruses from one environment and get them to grow on bacteria from another environment. Right, so that's Amiko. That's the only paper in the lab that had absolutely no revisions. It was accepted outright without even one comment other than publish it. We're like, you're fucking kidding me. All right, right, and then we know that they're moving important genes around. What's an example of an important gene?
NIF,
There's no NIF in those genomes.
PSPA.
PSPs, right? And what?
Or
Yeah,
is it-
so, that's the one
PSP.
that's easy. Probably exotoxins are the more classical one. right, and then, okay, and then we just have this thing, okay, so then what we're gonna do is we're gonna go into the human stuff, okay? And this is, I think most of you know this, this was really done, the collaboration is Jeff Gordon's lab, which is the guy that, like, he's been doing G proteins and stuff like that in the colon forever. Like, you know, how do we interact with our gut? How does our gut interact with the environment? Okay, so he did G proteins and he did a whole bunch of signal transduction, etc., in the gut environment. And then he got a lady named Ruth Lay, who was from Norm Pace's lab, came to his lab. Okay. And when Ruth was working with Jeff, they started doing culture-independent stuff, okay, in the human gut, okay? And that's kind of what led to this breakout field of the human microbiome, okay? The other thing that was going on is that there was another doctor, so he's a doctor by training. There was another doctor named David Relman, who's up at Stanford, okay? And David had independently gone and worked with Tom Schmidt out Michigan and learned how